Okay, so welcome to this next video in which we are discussing the use of the glucocorticoids to treat asthma. Okay, so glucocorticoids can be used as a prophylactic treatment to try and prevent asthmatic attacks. Now, in people with medium severity asthma, you can give inhaled glucocorticoids, okay? Whereas people with very, very severe and persistent asthma, you can start to give oral glucocorticoids. But glucocorticoids wouldn't be used in someone with mild intermittent asthma attacks. Instead, we do give them a bronchodilator. We'd give them beta-2 agonists, short-acting beta-2 agonists, which can be used when and where, basically, when necessary. So when they're having an asthmatic attack, they can squirt the drug into their airways and it can open back up the airways. Okay, so salbutamol and terbutaline are used for that. And glucocorticoids are used in more severe asthma. Okay, right, as a prophylactic, whereas that's not a prophylactic initially. Okay, so we've seen that glucocorticoids are going to go into cells and they're going to bind to the glucocorticoid receptor, GR here, and they're going to cause the dissociation of the glucocorticoid receptor from this complex of other proteins. So let's colour in the glucocorticoid receptor since it is the star of the show. Okay, so we'll colour it in blue here. Now, what's going to happen is these glucocorticoid receptors are then going to dimerize together. Once you've freed a glucocorticoid receptor like this from all these other proteins, it can now dimerize with another glucocorticoid receptor which has also been freed from its associated proteins. So here are two glucocorticoid receptors, two GRs, which are both dimerized together. And they both therefore have glucocorticoid molecules bound to them here, one here and one here, okay, which is the reason that they are free. Now, what's going to happen? Well, basically, these glucocorticoid receptor dimers are now going to translocate from the cytoplasm into the nucleus, and in the nucleus they're going to activate the transcription of a whole bunch of genes, and they can also repress the transcription of genes, but mainly they're going to activate. Okay, so let me discuss pathways by which they can activate the expression of genes. Okay, so basically, let's talk about DNA, okay? So, if we draw DNA here, so this is a little bit of DNA, okay? So basically, upstream of all genes in the human genome, you have what is known as a promoter region. Okay, so let's say this is a gene in the human genome here, this little box of DNA here. Okay, now upstream of this gene, and I'll just colour in the gene in pink here. Okay, so upstream of this gene you will have a region known as the promoter region. Okay, so let's say this is the promoter region here. And this will not be involved in actually being translated itself, okay, so it will not be involved in actually producing the protein. So it's a non-coding portion of DNA, but it's hugely involved in controlling the expression of the downstream gene. Okay, so basically the reason it controls the expression of the downstream gene is that the promoter region is where the enzyme RNA polymerase II has to bind. Okay, so RNA polymerase type 2 will come in and bind to the promoter region, and then it will work its way along the downstream gene. It will work its way along the DNA, it will transcribe the gene into mRNA, and then that mRNA will be translated into protein, and therefore you've produced the protein. So, the affinity of this promoter region for binding to RNA polymerase controls how often the RNA polymerase type 2 will bind to this promoter region, and therefore how much transcription of the gene you're going to get. So the promoter region does control the expression of this gene. Now, glucocorticoid receptor dimers, okay, can act as a transcription factor. Now, transcription factors are things which bind to these promoter regions and alter the affinity of the promoter region for binding to RNA polymerase type 2. Okay, so this is the transcription factor. So, basically you can have transcription factors which activate and also transcription factors which repress. So transcription factors which enhance the expression of the gene, they are going to bind to the promoter region of that gene and increase the affinity of that promoter region for binding to RNA pol 2, and therefore RNA polymerase type 2 is going to bind to that promoter region more often and then work its way down the downstream gene more often, producing a greater amount of mRNA, and therefore that will lead to a greater production of the protein. You can also have transcription factors which are repressors, and these will bind to the promoter region and make the affinity of the interaction between the promoter region and RNA polymerase 2 lower, basically, so they'll reduce the affinity, and therefore RNA polymerase type 2 will bind to the promoter region less often, and therefore you'll get less transcription of the gene, producing less mRNA, and therefore you'll get a reduction in the amount of protein that you're actually producing for this gene. Okay, so glucocorticoid receptors, therefore, can act as transcription factors, and I also want to stress that it's not as if a transcription factor is either an enhancer or a repressor. It doesn't work like that. It's not like you have one transcription factor and it will bind to the promoter region of all its genes and it will enhance the transcription of them. It can enhance the transcription of some and repress the transcription of others. So it depends on the promoter region, basically, and all genes will have their own little promoter regions. So a transcription factor will be a promoter at some genes and a repressor at other genes, okay? These glucocorticoid receptors, they can bind to promoter regions in front of certain genes, and some genes they'll upregulate and some genes they'll downregulate. Now, this is one mechanism by which they can upregulate and downregulate the expression of genes. There is another mechanism that I want to talk about by which they can upregulate the expression of genes. So, this involves histones, basically. So let me talk to you about the structure of a histone, basically. So, histones then. So, have I got space to do this here? Okay, I'll have a go. Right, so it's a very important concept in genetics. Basically, DNA is not just loose within the nucleus. Instead, it is wrapped around certain proteins very tightly. Okay, so let me show this. Right, so this is a histone protein that I'm drawing here. Okay, right, now histone proteins are octamers. They consist of 4 separate proteins. Let me try and show this. So here are the 8 separate proteins. Octamer is 8. They consist of 8 separate proteins. 1, 2, 3, 4, 5, 6, 7, and there's one that we can't see that will be somewhere back there. Okay, right. So, 2 of these proteins are what are known as H-free proteins. So they are histone-free. Okay, and I should write that word somewhere. So these are histone proteins, and they're going to form an histone octamer. Okay, so you have 2 histone-free proteins. Then you have 2 histone-4 proteins. So here is the 2 histone-4 proteins. And this structure that I've drawn so far, let me outline it, consisting of these 2 histone-3s and these 2 histone-4s. This is known as an H-free, histone-free, histone-4 tetramer. Okay, so this structure in green, this is distinguished from the 4 histones that are behind it. So this is what's known as the H3 H4 tetramer, and it's going to join on with a whole bunch of other histones to make the full histone octamer. Okay, so what we've drawn so far is what's known as the H3 H4 tetramer. Okay, now behind the H3 H4, what you're going to have then is more histones. Okay, so you're going to then have H2A and H2B histones. So let me show these. So behind H3 over here we'll have histone 2B. Okay, so this is histone 2B, and then here we'll have histone 2A. Okay, and then you'll have the same thing underneath. So this will be a histone 2A, and then the final one that we can't see, that's underneath this one here, that will again be a histone 2B. Okay, now these structures are divided up as well, but they're not divided into tetramers. Instead, they're divided into dimers. So this structure here, consisting of a histone 2A with a histone 2B, is what is known as a histone 2A. Let me put this down here. Histone 2A, histone 2B dimer. So basically, the histone octamer is made up of the H3H4 tetramer, where you have two H3s and two H4s joined together, and then it's made up of two H2A and H2B dimers. So you make two H2A, H2B histone dimers. You make one H3H4 tetramer. You join all of these together to get a histone octamer like this. Now, what's the significance of these histone octamers? Well, basically, DNA is going to wrap around this histone octamer like this. So, let me draw another picture of this, and I'll do it over the other way. Okay, so that I can show the DNA wrapping around the histone. Okay, so let's draw this again. So, here is our histone here. And we've discussed that the histone is made up of these 8 separate histone proteins. Okay, 2 histone 3s, 2 histone 4s, 2 histone 2Bs, and 2 histone 2As. Okay, so here are these 8 separate proteins. Right, now I'll relabel them up since we're only just getting familiar with this. Okay, so these two here, these are histone 3s, okay, so this is H3. These two here, this one and this one, these are 2 histone 4s, okay, and these two here, these are 2 histone 2Bs, and finally this one up here and the one beneath it, which we can't see, are histone 2As. Right. Okay, so DNA is going to wrap around these histone proteins that you have within the nucleus. So you don't just have DNA on its own, basically. It's wrapped around these histones. And let me show you this. So the DNA is going to start here, and it's going to wrap itself around the histone, like so. Okay, whoops, let me get that up there. Okay, and it's going to wrap itself around twice. So it's wrapped itself around once now. Okay, and it's going to wrap itself around again. Okay, and it's going to come out like this, and then it'll go off. Okay, so here's the start, here's the finish. So it wraps around twice. It wraps around the histone twice. Okay, and this structure where you have the histone octamer with the DNA wrapped around it. And remember, this is a double strand of DNA, so this blue line means the double helix, basically. It means one of these structures. So the blue line means this, okay? So we were just denoting this entire thing as this blue line, okay? Right, so the whole double helix is wrapped around the histone octamer like this, and this is what's known as a nucleosome. Now, basically, when the DNA is coiled very tightly around this histone octamer in these nucleosomes, it's very difficult to get RNA polymerase 2 to come in and bind to the promoter regions, basically. Very difficult indeed, because there's just no space for it to bind, basically, because the DNA is wrapped so tightly around the nucleosome. So you need to loosen the binding of the wrapping of the DNA around the histone octamer, okay? And the way that you can do that is by acetylating certain residues on the histone, so by adding acetyl groups onto certain residues on the histones. And this is another thing that these glucocorticoid receptor dimers are going to trigger. They're going to trigger histone acetylation. Okay, so where they have bound to promoter regions, okay, so we've got these dimers of glucocorticoid receptors bound to promoter regions. What will happen is a complex will assemble on top of these. So let me show this. Okay, so here are the two glucocorticoid receptors here. Okay, and what's going to assemble on top of them is basically a complex which is going to function as a histone acetylase, or a histone acetyl transferase is the correct thing. Sorry, not a histone acetylase. A histone acetylase would be an enzyme that removes acetyl groups. Okay, so let's put the DNA here. So it's bound to the DNA, it's bound to the promoter region of the DNA, and basically we now want to assemble a histone acetyl transferase, as it's called, which is an enzyme which adds acetyl groups onto structures. Okay, so what's going to happen is the glucocorticoid receptors are here, and now what's going to happen is another protein is going to bind on top of these. Okay, and this next protein is what's known as SARC. Okay, and SARC stands for steroid receptor coactivator 1. Okay, so it's specifically SARC1 we're dealing with here. So I'm going to put that little 1 in there. So this is steroid receptor coactivator 1. So steroid is for S, receptor is for R, C is for coactivator, and then it's the steroid receptor coactivator 1. Okay, now on top of the steroid receptor coactivator 1, two other proteins are going to bind. So let me show these here. Okay, like so. And these two other proteins, one is known as CBP, which stands for Krebs binding protein. So I'll put that down here. So CBP stands for Krebs binding protein. Krebs is another very important transcription factor. Okay, so this is Krebs binding protein. And then the other protein that's going to bind here is what's known as PCAF. PCAF. Okay, and PCAF stands for P300, and you'll see why in a moment. P300 CBP is the C there. So P300 Krebs binding protein, and then associated factor. So this is the P300 and CBP associated factor, or PCAF for short. Okay, and on the surface of this Krebs binding protein and P300 CBP associated factor, you're going to bind then P300. Now this complex is then what's known as a histone acetyl transferase. So this is an example of a histone acetyl transferase, or a HAT basically. And these HATs, these histone acetyl transferases, are going to add acetyl groups specifically onto lysine residues in the histone 4 protein. Okay, so let's now discuss these lysine residues in the histone 4 protein. Okay, so let me discuss the structure of a lysine residue. Okay, so lysine is an amino acid which you have within proteins. So I'll draw it as though it's a residue, i.e. that it's as though it's actually in a protein rather than as the pure amino acid. Okay, so here is the core amino acid structure, the amino terminus, the alpha carbon with a hydrogen off it, the carboxylic acid group that will be bound to another amino group down here, of the next amino acid along. Now the R group of lysine is that you have 4 methylene groups, and because I'm lazy I might just draw 1 methylene group, put brackets around it, and then stick a 4 down there. And then on the end of that you then have an amino group. Okay, so this is the normal structure of a lysine amino acid residue, and the single letter amino acid code for lysine is K. Okay, now basically you can add acetate or acetyl groups onto this amino group of the lysine. Okay, so let me show you the structure of acetic acid. Okay, so here's the carboxylic acid group, and then you just have a methyl group off like this. So this is the structure of acetic acid. It's what would now be called ethanoic acid, and it's what's in vinegar basically. Now you can form an amide link between this amino group here and this acetic acid molecule. You can take off the alcohol group from the carboxylic acid molecule, a hydrogen from the amino group, and then you can link this carbon here of the acetic acid molecule to the amino group of the lysine residue, and that produces you an acetylated lysine, okay? So you can add acetyl groups onto lysines. Now, this histone acetyl transferase enzyme that we've produced is going to add acetyl groups onto lysines that are on histone 4 proteins here, okay? And it's specifically going to add acetyl groups onto lysine at position 5 and also lysine at position 16. Okay, and when you add these acetyl groups onto lysine at position 5 and lysine at position 16, this loosens the coiling of the DNA around the histone octamer, okay? And that means that RNA polymerase can now gain access to the promoter region, whereas before it couldn't. So, both the direct transcription factor activity, where you're binding to the promoter region and altering the affinity of the promoter region for RNA polymerase 2, and also the fact that we are modifying the histones so that the DNA doesn't coil quite so tightly around the histones. Both of these can alter the likelihood that RNA pol 2 will bind to the promoter region, and therefore the likelihood that the downstream gene will be transcribed. So this is a mechanism for increasing the expression. Of course, if we're trying to repress the expression, then this isn't a valid mechanism, but this is another way that glucocorticoid receptor dimers are going to increase the expression of certain genes.